All right, in this little series on metabolism, I wanted to start off with this video, an introduction to metabolism, um, with this statement. Metabolism is a story of carbon oxidation. Now, what does that mean? I hope that in this introduction to metabolism, we'll have a better understanding of what that means and why it's important to understand where the energy comes from that we use to produce ATP. I started this video with this little picture of a campfire, and we see wood burning. This doesn't surprise us. Everybody knows wood burns. Now, from a scientific point of view, I think it's important to recognize that that wood is primarily cellulose, which is glucose, right? So in many ways, the fact that that fire is burning is very, very similar to exactly what's going on in our bodies when we eat glucose. But a small wood burning fire to me is a little bit boring. I am much more interested in knowing what happens if we take something like methane and we dump it on that fire. We expose everyone's favorite gas, methane, to that fire. And what do you get? Boom, you get an explosion, right? This is not just fire, this is violent fire. A release of energy that is significant and felt in terms of heat and force. Now, I don't want to go into the details of Gibbs free energy and spontaneity and thermodynamics, things like that, that actually describe the process. But I do want to start by simply drawing a picture of methane, the simplest hydrocarbon, CH4, and trying to understand what happens to it when it burns. The end product we know will be what? Carbon dioxide. Here's carbon dioxide over here. I want just for a second for you to take a look at these two molecules and tell me what the difference is between these two carbons. The carbon in here in methane versus the carbon over here in carbon dioxide. And if we understand the difference between those two carbons, we'll understand this concept of carbon oxidation. Now let's define oxidation and reduction. The easiest and simplest way to remember this is the mnemonic oil rig. Not that oxidation and reduction reactions have anything to do with oil or boats, but this helps us remember what this stands for and helps us make sure we don't get confused. Oxidation is loss, while reduction is gain. Hopefully your question is loss or gain of what? And the answer to that is I'm not going to tell you. But I do want to come down here. Let's do a little Pictionary. What we come up with? It's a battery. Everybody knows that's a battery. I want to talk for just a second about how the battery works. Whether this is a C-size battery, a AAA, doesn't really matter. For all of those with this overall general architecture, this battery has a rating of 1.2 volts. And I'd like for just a second to think about what that means. Batteries are super important. They power our devices, our phones, our computers. It starts our car in the morning. Batteries are essential for daily life for you and I. And right here, we have a battery. And it says 1.2 volts. What does it mean? Well, what do we know about batteries? We know from life experiences that a battery has two sides, a positive and a negative. There's the negative, there's the positive. When we connect these, if we were to connect these with a wire, maybe we hook a voltmeter up to it, so we put a little reader on it, and we connect that, and then we have this little needle right here, and it tells us, it points, oh, 1.2 volts, perfect. What's it measuring? Volts describes potential. It doesn't describe the flow of current, but rather the desire the electrons have to flow, right? And in a typical battery, all you really have are two different ions that have different electronegativities. One wants electrons more than the other. And so when we think about batteries, it's the flow of electrons that gives us power. Now, and this is really important to recognize, I didn't say it's the electrons that gives us power. The electrons they're just electrons. But when they flow, now we're able to take potential energy, convert it into kinetic energy, and that kinetic energy powers the world. It turns on the lights. Like I said, it's in your phone, it's in your computer, it's everywhere. The flow of electrons is power. With that in mind, let's come back up here and see if we can now understand oxidation reduction. Oxidation is loss of what? Of electrons, while reduction is gain of electrons. Now, because oxidation and reduction typically are happening at the same time, something is losing while something else is gaining, we combine this term and we come up with the term redox. A redox reaction is simply a reaction in which we see a transfer of electrons from one thing to another. There's a verb there, transfer. Please associate that with what we were talking about, flow. If I'm moving electrons, I can use that flow of electrons eventually to do work. Now, what does this have to do with metabolism? Well, remember, metabolism is the story of carbon oxidation. As we go back and we compare these two molecules that we have, we've got carbon dioxide, we've got methane. Do you now see the difference between the carbon in methane and the carbon in carbon dioxide? Methane, very high potential energy. Why? Because this carbon is reduced. We compare that with carbon dioxide. This is low potential energy because that carbon is completely oxidized. Now let's see if we can understand what the difference is. Hopefully by looking at these two pictures you can see one, we've got hydrogen, the other carbon is bound to oxygen. Let's walk this kind of through a step-by-step process. First of all, we know that oxygen is more electronegative than carbon, correct? Whereas hydrogen is about the same. Now, in this molecule above, in methane, how do we make it? Well, we've got carbon. We know carbon has 4 valence electrons. Here's carbon's electrons. Along comes hydrogen. And hydrogen is going to be here and here and here and here. There's a Lewis dot structure showing methane. In these cases, look at that bond. How would you describe that bond? Because the electronegativities between carbon and hydrogen are relatively similar, then we're going to call this a non-polar covalent bond. Non-polar because they are shared equally. But if we contrast that with a bond between carbon and oxygen, we know that oxygen has a greater pull on those electrons. Oxygen has a higher electronegativity and is essentially pulling that electron away from carbon. So with that as our background, what if we were to oxidize one of the 4 electrons on this methane? Right? What's the next step? It's methanol, where I have carbon, and it's still bound to hydrogen, 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 but now it's bound to a hydroxyl group, an alcohol group, OH. There's methanol. What do we see? We see that in this bond, right here, it's unequal sharing. carbon has lost that electron. Remember, oxidation is loss of electron. So that represents a higher oxidation state for carbon. Where does it go next in the journey? Well, we've got a carbonyl group, a carbon double bonded to an oxygen. This carbon has 2 electrons lost. Now you can begin to see the importance of this role that oxidation plays in metabolism. The next step is the carboxyl group. We have a double bonded oxygen here with an OH over here. This represents carbon that is nearly fully oxidized. It only owns one of its electrons. And then finally, where do we end up? As carbon dioxide. Of course, the explosion that we have from methane, it didn't go through all of these steps. It went directly from methane to carbon dioxide. But for the human body, we take it step by step so that we don't lose a massive amount of energy as heat. For each little step, we extract energy and we use that energy eventually to make ATP. I want to highlight exactly how this is done with an example using pyruvate. Pyruvate, of course, is the end product of glycolysis. We'll talk about glycolysis in a different video. Here's pyruvate. C, it's got 3 carbons, C double bond O, C double bond O, OH. This is pyruvate. Now, in order for pyruvate to enter the Krebs cycle, it's first got to be oxidized into acetyl-CoA. Remember, each one of these bonds that hold this molecule together represents a pair of electrons. And I want to follow the electrons. I also need to point out all of these steps are done by enzymes. Enzymes regulate the kinetics of these reactions. For this reaction to happen, let's bring in a coenzyme. We get coenzymes. These are the vitamins that we eat, and coenzymes, I want you to think about them as transporters. They're really good at carrying things around. Coenzyme A works through a functional group that is a sulfhydryl group, so we're gonna throw that S out there. Sulfurs are really good at grabbing things, but also letting things go. So what we're going to do, we're going to bring these electrons that sulfur has, and we're going to use them to bond to that carbon. Now, in terms of counting carbon, I want you to recognize this. This sulfur is actually more electronegative than the carbons that are currently bound to this central carbon. And so this bond right here that we just drew is a more stable bond. It's a bond that this carbon would rather have. Now watch what happens. Let's take these electrons here and let's swing them over here and put them right there. We've just broken that bond and move those electrons to have it double bonded to oxygen. Again, this is a spontaneous thing simply because oxygen is more electronegative than carbon. So if I can take a carbon-oxygen bond and convert it into a carbon-carbon bond and convert it into a carbon-oxygen bond, it's more stable. It's at a lower energy state. And this is very much in line with the second law of thermodynamics. Everyone's seeking the lowest energy state of stability. But now, importantly, I want you to see what happens. This oxygen here is now making too many bonds. So what happens? This hydrogen, but not just the hydrogen and its pair of electrons are kicked off. So we have a hydrogen, 2 electrons, it's ionized as a negative ion. This is called hydride ion. Now, as part of this chemical reaction, we have another coenzyme, NAD+. This is a vitamin B3 derivative. NAD+ comes over and picks up that hydride ion, its hydrogen and the 2 electrons. And what do I get out of it? I get NADH. I hope you can appreciate what we just did. Pyruvate was just oxidized. I took electrons off of pyruvate, and where did I put them? I loaded them onto this NAD+ molecule to make NADH. So NADH represents the reduced form of this molecule because it's carrying extra electrons. And what else did we get out of it? Well, we can see the acetyl-CoA that's now ready to enter the Krebs cycle, and we also recognize that this molecule right here came off as carbon dioxide, the most oxidized form of carbon. And what do we do with it? We breathe it out, right? We've extracted all the energy there is in that carbon. It's no good to us anymore. And so we exhale it as a metabolic waste product. Now, I want to come back to finish our story to the electrons, because that's what we've been following. NADH has those electrons loaded onto it. Let's go back to our battery. NADH doesn't really like those electrons. It's a good transporter because it doesn't like them too much. It's like, okay, yeah, you can join me, but I don't have a strong affinity for you. NADH therefore represents the negative terminal on this battery. In fact, mathematically, we can do some calculations and we can see that it actually has a reduction potential, which describes its desire for electrons of a negative 0.32 volts. Now we need the positive end of the battery. Where do these electrons really, really, really want to go? Going back to the very idea of breathing, this is what oxygen gas is for. Oxygen, because it's so electronegative, really loves electrons. In fact, the reduction potential for oxygen at 0.816 volts, you can see that's much, much higher than NADH. With those values, we can actually now calculate the voltage of the electron transport chain inside every single cell in our body. That's right. Every cell in the body has its own little mini battery. And we use that battery just like we use batteries today to do work, to power things. We can calculate the voltage of this battery. The overall voltage is just going to be the difference between these reduction potentials. 0.816 minus a negative 0.32 gives us an overall 1.136 volts for the mitochondria in each of our cells. And what am I going to do with the battery? I'm going to make ATP. So let's go back and see if we can summarize this. We started by saying metabolism is a story of carbon oxidation. Now we understand what that means. What we're going to do is we're going to take the food that we eat and we're essentially going to strip the energy off of it, just like a fire is stripping the energy off of that wood or methane. Whether it's carbohydrates or proteins or fats, all of these are organic carbon-based molecules, and the carbon in those molecules is in a reduced state. As I eat them, the body takes those macromolecules and runs it through the steps of glycolysis or beta oxidation or Krebs cycle, whatever it is, and we'll talk about those individually later, but those are the processes used to strip electrons off of carbon. And when I strip those electrons off of carbon, I can run them through the electron transport chain, which is shown here by this battery, where we start with NADH or FADH2, and we end up on oxygen as our terminal electron acceptor. The journey, the flow of those electrons from NADH to oxygen provides power. And with that power, the cell can make ATP, which is the currency of the cell that the cell uses for nearly all of its energy demands.